Protein enzymes are biological catalysts, meaning in the cell, these molecules increase the chemical reaction rate, without itself being changed. Naturally, a bottle of hydrogen peroxide to decompose into water and oxygen will take many months. A non-catalyzed reaction requires above 450 Celsius degree temperature heat and 200 times atmospheric pressure for this reaction. With adding iron catalyst, the reaction will increase 1,000-fold. With enzyme, the rate of chemical reaction increase up 1 million to 1 billion-fold. Enzyme can perform this action in normal temperature of a living organism and with much high rate. Most enzymes have names that end with ase. The few exceptions include some of the first discovered enzymes for digesting protein, such as pepsin, trypsin, and chymotrypsin. The rest of enzyme names tells you something about what the enzyme does. Enzymes are classified into 6 groups: oxidoreductases, transferases, hydrolases, lyases, isomerase, and ligases. Oxidoreductases catalyze oxidation-reduction reactions. Oxidoreductase enzymes are a group of enzymes that catalyzes the transfer of electrons from one molecule, the reductant, also called the electron donor, to another, the oxidant, also called the electron acceptor. Oxidoreductase enzymes utilize NADP+ or NAD+ as cofactors. Oxidoreductase enzymes include the following subgroups of enzymes: oxidase, dehydrogenase, peroxidase, hydroxylase, oxygenase, and reductase. Transferases catalyze transfer of functional groups from one molecule to another. Systematic names of transferases are constructed in the form of donor, acceptor group transferase. However, other accepted names are more frequently used for transferases, are often formed as acceptor group transferase or donor group transferase. In the Enzyme Commission (EC) system of classification, the accepted name for RNA polymerase is DNA-directed RNA polymerase. EC numbers are assigned by the Nomenclature Committee of the International Union of Biochemistry and Molecular Biology. Hydrolases catalyze hydrolytic cleavage. Hydrolase is a class of enzyme that commonly perform as biochemical catalysts that use water to break a chemical bond, which typically results in dividing a larger molecule into smaller molecules. Some common examples of hydrolase enzymes are esterases, lipases, phosphatases, glycosidases, peptidases, and nucleosidases. Hydrolase enzymes are important for the body because they have degradative properties. Lyases are enzymes cleave bonds in such manner that the cleavage event does not involve hydrolysis or oxidation. During cleavage, a molecule is eliminated from the substrate, leaving an unsaturated residue. The decarboxylation of acetoacetate 2, acetone and carbon dioxide is a familiar example of a lyase reaction, in which a C-C bond is cleaved. Isomerases catalyze intramolecular rearrangement. Isomerases are a general class of enzymes that convert a molecule from one isomer to another, meaning the end product has the same molecular formula, but a different physical structure. Ligases catalyze reactions in which two molecules are joined. Ligase is an enzyme that can catalyze the joining, ligation, of two large molecules by forming a new chemical bond. Other common names for ligases includes the word synthetase, because they are used to synthesize new molecules. Enthalpy is the heat content of a system. As we all know, the heat can go in or out of the system. If this system is a chemical reaction, the change of heat is called enthalpy change, as enthalpy is a state function, and it is dependent on the changes between the initial and the final state. Endothermic reactions are those in which there is an absorption of heat from the surroundings environment. Here, the enthalpy of the products is higher than the enthalpy of the reactants, and the enthalpy change is positive. Exothermic reactions are those in which there is a release of heat, and that energy is given out to the surroundings. In this type of reaction, the enthalpy of the products is lower than the enthalpy of the reactants, and consequently the enthalpy change is neg. Entropy refers to the measure of the level of disorder in a thermodynamic system. There is a tendency in nature for systems to proceed toward a state of greater disorder or randomness. the entropy of a room that has been recently cleaned and organized is low. As time goes by, it likely will become more disordered and thus increasing the entropy, until large amount of energy is used to bring it back to order. So, for any spontaneous process, the entropy of the system should increase. Entropy is denoted by S. Spontaneous reaction is considered to be natural, because it is a reaction that occurs by itself, without any external action towards it. Non-spontaneous reaction needs constant external energy applied to it, in order for the process to continue, and once you stop the external action, the process will cease. If change of G is negative, then it's spontaneous. If change of G is positive, then it's non-spontaneous. On the reaction path, between the initial and final arrangements of atoms or molecules, there exists an intermediate configuration, at which the potential energy has a maximum value. Yes, you heard it, the potential energy. The configuration, corresponding to this maximum, is known as the activated complex, and its state is referred to as the transition state. The point of highest free energy is called the transition state of the system. Activation energy is the minimum energy that a reacting or flowing system must acquire for the transformation to take place. Catalysts act by providing a reaction pathway with a transition state whose free energy is lower than that in the uncatalyzed reaction. The difference between the values of activation energy G for the uncatalyzed and catalyzed reactions indicates the efficiency of the catalyst. Note that a catalyst lowers the free energy barrier by the same amount for both the forward and reverse reactions. Consequently, a catalyst equally accelerates the forward and reverse reactions. The difference between these two energies is the change in Gibbs free energy during the reaction. The net change in free energy is negative in the conversion of S to P, as energy is released by the reaction when S is transformed to P. The line increases from the energy level of the substrate, indicating that some energy input is needed before the reaction proceeds downhill. How does the enzyme make it easier to reach the transition state? Well, enzymes have an active site, a pocket into which the substrate fits, and where the chemical reaction occurs. The pocket is created by the 3D folding of the polypeptide. This fashion of enzyme-substrate association can be explained using a lock and key analogy, which was first postulated in 1894 by Emil Fischer. Some enzymes require no chemical groups for activity, others require an additional chemical component, called a cofactor, either one or more inorganic ions, such as iron ion, magnesium, manganese ions, or zinc ion, or a complex organic or metal-organic molecule, called a coenzyme. Some enzymes require both a coenzyme and one or more metal ions for activity. A coenzyme or metal ion that is very tightly or even covalently bound to the enzyme protein is called a prosthetic group. A complete, catalytically active enzyme together with its bound coenzyme and or metal ions is called a holoenzyme. The protein part of such an enzyme is called the apoenzyme or apoprotein. Enzymes have serious consequences for your health. There must be something that senses the conditions, and then a way to switch an enzyme on or off. When an enzyme active site is covered, until needed, then such enzymes are called Zymogens or proenzymes, for example prolipase or trypsinogen, or chymotrypsinogen. From the name it tells that these enzymes are inactive. These enzymes become active by active site bond cleavage. Some enzymes requires phosphate for activation and inactivation. For example, glycogen phosphorylase gets active when phosphate is attached, whereas glycogen synthase gets active when phosphate is removed from it. Special phosphate-adding enzymes are called kinases, and special enzymes that remove the phosphate group from enzymes are called phosphatases. There are certain family of drugs that binds with active site of enzymes, thus inactive enzymes. These drugs are called enzyme inhibitors. Such drugs that permanently inactive the enzymes, also known as irreversible inhibitors. These drugs are also called suicide inhibitors, which covalently bond to the enzyme, and making them inactive permanently. Reversible inhibitors has non-covalently interactions with the enzymes, making them inactive temporary, and they can be removed most probably with dilution or dialysis. The enzymes become active again. The reversible inhibition undergoes in 5 different kinds. 1. Competitive inhibition. 2. Non-competitive inhibition, or allosteric inhibition. 3. Partial competitive or mixed inhibition. 4. Uncompetitive. 5. Feedback inhibition. Competitive inhibitors molecules resemble the normal substrates, taking over the enzyme active site, thus directly competing with the substrate in a reaction. However, a competitive inhibitor can be overcome simply by adding more substrate. The inhibitory molecule in non-competitive inhibition does not bind to the active site, rather to another site of the enzyme called allosteric site. This brings slight change in conformation of the active site, though the substrate can be bound to the active site, but temporarily altering the enzyme activation. In this type of inhibition, the inhibitor molecules, when attached to allosteric site, it somehow mess up the binding site, hence preventing substrate binding with the active site of enzyme. This inhibition by many has considered it as non-competitive inhibition. In this inhibition, after the binding of substrate to the active site, it induces a conformational change in the allosteric site, allowing the inhibitor to bind, which interferes with the machinery of the enzyme, resulting in inactivation of enzyme, though the substrate is bound. In living organisms, metabolism is regulated in a series of reactions, involving many enzymes for anabolism or catabolism of compounds. For example, bacteria synthesize isoleucine from threonine in a series of 5 enzyme catalyzed steps. In this process, the final product inhibits the enzyme that catalyzes the first step in a series of reactions. As the concentration of isoleucine increases, some of it binds as a non-competitive inhibitor to the first enzyme of the series, threonine deaminase, thus bringing about a decrease in the amount of isoleucine being formed.